Hi, everybody. It's just going to be me narrating. This is the review for the exam. I'm just getting it cooked up. All right. So the exam is 3 chapters, so it's kind of long. It's just 70 questions. Uh, it is the cell stuff. To be fair, is probably should all be reviewed for everybody. And the ones where I think a fair amount of the time or a fair amount of the material comes from that neuromuscular junction and basically the muscle physiology unit and now the nerve physiology unit. It's not in a specific order. I've sort of blended it. So, for instance, I'm going to just kind of as I see them, as I look at the exam and talk about it. I mean, we start with the neurotransmitters. And thankfully, that should be as fresh as anything. It's the most recent thing we did, just about. And so there, understand it's the categories. And there's important things in the categories. I mean, the one we know the most about is acetylcholine. I shouldn't have to do a whole lot of review from that. We know it's neuromuscular junction. We know it's in the autonomic nervous system, which interestingly is not on this exam, but you're going to get to that later. So you know it's there, and it's certainly in the brain. That's unique in of itself. You have the biogenic amines. And again, what's important about the biogenic amines is where they come from. Remember, epinephrine, norepinephrine, and dopamine come from tyrosine. Tyrosine also makes thyroid hormone, so it's pretty important. The indolamines, you have histamine from histidine, and perhaps most importantly, serotonin. Okay, and serotonin comes from that tryptophan. And so indole is just you're probably familiar with it from for those who've had biochemistry as part of it. But I mean, these are really significant areas. When we talk about catecholamines, that was really the term for epinephrine, norepinephrine, and dopamine. They fell into that category. Those ones that were derived from tyrosine, they're typically, with very, very rare exceptions, excitatory. Serotonin, histamine, depending on the receptors, they can be either excitatory or inhibitory. Okay. Don't forget those endogenous morphines, the enkephalins. Those are those sort of opioid-like. In other words, they're very similar chemically to the nucleus of morphine. Sort of, people have talked about them when they talk about endorphins or enkephalins. These things that are painkillers that we produce, or things that trigger pleasure centers to be able to, sometimes from certain physical activities, and particularly for overcoming pain and certain things like getting it from a good workout, let's say. And a lot of people really, there's almost a positive kind of addiction that develops for those kinds of physical activities unless they get overwhelmed by it. And then the one we probably know the least about are these gaseous agents, and truly nitric oxide is the most interesting. We're learning so much about it as its use in growth and development, uh, the impact it has on blood vessels and dilation, uh, almost being produced in such a way that it's almost auto modifying, sort of auto controlling our blood pressure at even a small blood vessel level. Very interesting. And of course, down the road we'll talk about its role, vis-a-vis interacting with it for ED or erectile dysfunction medication. So that's just a laundry list. So know a little, what I call a nugget, or little factoids, little interesting facts about all of those that I went over, and you can listen to that certainly that recording where I covered in some length. We know the distinction, know how the G protein receptors work. You should have had this 63 times by now. That's the primary messenger. They don't get into the cell. Their actions are indirect. They use cyclic AMP and sometimes ionic calcium as second messengers. That's pretty consistent with regard to those things. Those are the ones we see the most when you're talking about steroid based receptors, or fat soluble based hormones, rather. And effectively, those targets are located in the cytoplasm, sometimes in the nucleoplasm within the nucleus. And basically, the receptors are there. They form a complex with the receptors, eventually making their way into the nuclear area and having their effects not through second messengers, but directly altering transcription and translation. And you see this again and again. We went over the cell cycle. You know all those parts. You must have had it 16 times by now. The phases, what goes on in each one, where the checkpoints are. Remember, G1, S, and G2, they are G1 is surely the most active. Everything is replicating. Basically, building all the building blocks are replicating, not so much assembling, but replicating. It's a very, very busy stage. Effectively, the waste products or byproducts create those cyclins. They bond with cyclin-dependent kinases. Those chemical agents, those enzymes, those chemical agents are produced, and then complexes form when there's a sufficient amount of cyclin-dependent kinases, cyclins, and the kinases they bond to it. Effectively, it releases the stop, okay, the limiting factor at the checkpoint, and it allows it to proceed to S phase. S phase is where we see DNA, okay, and where we see DNA assembled. So if you're going to get a mutation, an error in DNA, that's where it occurs, period. That's the big job in S phase. And then in G2, it's like a final check. Now we've replicated the DNA. We've replicated all the building blocks. We started to assemble various very intracellular and membrane type components. And again, there's an abundance of materials in the form of cyclins that are produced, meeting up with the cyclin-dependent kinases. If they're sufficient, boom, they release that so-called G2M checkpoint, and it proceeds from that point on to mitosis and cytokinesis. The reality is, okay, that when there's defects, the remedy for the most part is apoptosis. Sometimes they'll exit the cell line in G1 when they have developed or differentiated sufficiently. We call that G sub 0. Think of a finally developed muscle or nerve cell. We might think of those happening more in gestation than necessarily being part of development, but there's some with regard to development as well. And then subsequent to that, okay, yeah, apoptosis, particularly in the second phase, in the G2 checkpoint area, because it's almost a surveillance system with the likelihood of a mutation or a significant mutation could really alter the cell function. So, I mean, those are just little things to keep in mind. So, I mean, I think you need to look at all of those things as well, particularly the role of the cyclins and the cyclin-dependent kinases. We've gone over that more than once, I'm sure you have. And then all of the events, I mean, this is mitosis 101, the 4 events that are associated with prophase, released in telophase. Anaphase is the shortest. They pull apart, metaphase. They line up down the sort of the equatorial line of the cell. We're all familiar with that. Not going to rebuild the wheel here. What are the 4 things? The nucleolus disappears. The cell membrane disappears. They don't really disappear. They sort of decompose. In a sense, they disassemble and they're no longer visible. And we see the spindle begin to assemble, and certainly the chromosomes form. They're revealed. They go from chromatin as they begin to supercoil and become chromosomes, and we reverse all of that in telophase. Cytokinesis can occur as early as anaphase. Typically, it's during telophase. And cytokinesis is cellular division, while mitosis is nuclear division. That's a really important point. That's why read the darn questions. You know about the organelles. I'm not going to go over. It's a mitochondria, and it has 2 membranes in the inner membrane and the cristae, and what it does, and lysosomes, enzymes for digestion and defense, and peroxisomes, basically defensive, toxic derivatives of oxygen, etc., etc. You know, you've got to have all those already in your toolbox, I think. Okay, so all of the things that it does, okay, are really important. We know where things are produced. When you're talking about peroxisomes, lysosomes, when you're talking about exocytosis, we're talking about membrane proteins, all of those things, that's the definition of what goes on in the rough endoplasmic reticulum. That's proteins that are either going to be bound in or incorporated in membrane. So they could be incorporated, they could be bound within membrane vesicles that may be in the cell, lysosomes, peroxisomes, vesicles that are exocytosed, secretory vesicles like neurotransmitters, or they could be proteins that eventually are incorporated in the cell's membrane itself and become any one of the number of membrane proteins with the various and sundry roles that they have. Remember, those are the kind of the three destinations of stuff coming from the rough endoplasmic reticulum. Where smooth is all about, I mean, other than processing lipids, storage areas, particularly with regard to muscle, both skeletal and smooth, when we're talking about intra and extracellular calcium, all that stuff. I mean, there's just a whole lot going on. And you've had all this before, so you should be able to do it. Okay? Those are all the different factors that are involved. We don't have to go over diffusion, osmosis, facilitated diffusion, blah, blah, blah. The big driver is concentration gradient. Interestingly, and we really see this in respiratory perhaps as much as anywhere, is we see the effectively the permeability involving the membrane being important. You're going to see that in respiratory. Oxygen has a fairly large concentration gradient when you're talking about from the lungs to the bloodstream. Carbon dioxide does not. It's relatively small concentration gradient. Why do they move equally? Carbon dioxide, as you will learn, is much more soluble. Okay, goes through the membrane much more rapidly. It's not even a question of receptors. It's a question of solubility. And again, I mean, we're not going to go. Solubility is a concept we all understand with regard to chemistry and things like that. But just some things can move faster through certain areas than other things, and this is one of the examples. You know all about the sodium potassium pumps. You know all about primary active transport. Basically, the sodium potassium pump is the classic example of that. A lot of times, that's really primary. It bringing in, triggering other pathways to allow glucose to enter the cell, secondary active transport. Moving molecules in the same co-transport or opposite direction, anti-transport. Another term for co-transport is symport. Okay, all of those are important. We know about exocytosis. We know about endocytosis. Endo as vago and pino. One's technically solids, one's liquids or oils. That's the pino portion of it. Normally, this is receptor driven. The classic, when we talk about very specific receptor driven or receptor mediated endocytosis, typically the proteins are called clathrin, CLATHRIN, coated pits, because they not only receive their receptors are triggered by a specific chemical or chemicals. It also induces the formation of that vesicle that's going to facilitate the endocytosis of it. Okay, and you should be aware. You should be really aware of that as well. We're going to deal with buffer systems. We talked about the carbonic acid buffer more than once in a variety of classes. We'll continue to do that again. So just be notable about that. The other buffer systems are proteins in the plasma have a buffering system. Calcium in the plasma and in the blood itself has its own unique ability to buffer. But the principal buffer and the brilliance of it all is to take two waste products, water and carbon dioxide, in the presence of an enzyme, carbonic anhydrase, and boom, reversibly be able to create carbonic acid, a weak acid. And what makes great buffers? Weak acids. Because they're weak. Strong acids give up their protons immediately, and sometimes so vigorously, toxically, burns, whatever you want to describe it, rather violently might be the way to think about it. The flip side of that, okay, is weak acids. Weak acids, if anything, the strong part's called their conjugate base. That's from chemistry, and it's not a chemistry class, but effectively, weak acid in the sense of what does it do? It will give up a proton or not. It's as likely to hold on to it as to give it up, and that's what makes a good buffer, its ability to be a proton donor or to be a proton acceptor. You know about snare proteins. God, you must have had that a bunch of times, V snares and T snares. The role that they play. Remember that synaptotagment, whatever it is, the name. I don't even think it's on the test, but understand the role it is. How that's interfered with the Botox at the neuromuscular junction and interferes with the snare proteins being efficient and being able to, and basically what Botox does is blocks the secretion of the acetylcholine. And see, where something like when we look, remember we talked about the disease that myasthenia gravis, that was an autoimmune disease affecting the receptors for the acetylcholine. A little bit different. You know, when we're talking about Botox, and they're both effectively paralytic, though obviously the myasthenia gravis takes time over a sufficient period of time to affect the neuromuscular junction. I mean, that we understand that a little bit better, let's say, than Lou Gehrig's disease, amyotrophic lateral sclerosis involving upper and lower motor neurons. Yes, they're motor neurons, but it's not clear why both of those systems are effective and why we lose control over that. Is it glutamate toxicity? There's a variety of things that they've talked about over time. I mean, eventually people will know more about it, and that means we'll be able to treat it much more efficiently. But that's not on this test either. I just comment on that as a side when we were looking at neuromuscular disorders. This is also the part we visit resting membrane potential, both in all of these chapters. So understanding positive on the outside, negative on the inside, the more important role, if you will, of sodium, only because there's more sodium leak channels than there are. The important role of potassium, because I beg your pardon, I misspoke. There are more potassium leak channels than sodium leak channels. Potassium really sets the resting membrane potential, and if anything, the sodium being attracted back in through its more limited leak channels being attracted in to compensate may offsets it, so it goes like to minus 90, then the sodium will bring it up to minus 70, and of course what continues to go on, whether it's a cell that does not have the potential to be excitable or is the constant usage of those sodium potassium pumps, because effectively it's an equilibrium. We will always have potassium leaking out and sodium leaking in. So those pumps will continue to have to work to restore the chemical basis, not so much the electrical basis, but the chemical basis. So effectively, to compensate for the sodiums that have been brought back in, we're going to have to spit some back out and bring some potassiums back in. Again, things happen electrically first, and then the chemical events. So electrical events happen first, then chemical events, and sometimes and often mechanical events, as you will see going forward. And we talked about the role of proteins and what they do. They attract water. Colloids is what they're. So you're familiar with all of these types of things from a biochemical basis. So let me see, making good progress here. So a lot of this is the sort of the elementary stuff, other than what I talked about with the. This is like what I call the cellular and basic physiology systems, with the exception of the neurotransmitter material. So now we get more into, in this next section of the test, focuses more on the neuromuscular junction, cross bridge cycle, excitation, contraction, coupling. You got to know the roles of tropomyosin, the thread-like protein, troponin, the plug-like protein. They're considered regulatory proteins. The role of actin, the role of calcium, the role of titin, that little elastic protein, the myosin, which is really a true motor protein, because motor proteins effectively they change shape to create movement. That'd be it. They're not. They don't all move things the same way, but the fact that they change shape to create movement, I think, is a very good working definition of a motor protein. So remember the whole deal with it. All that sequence of event. What happens? The nerve going to the muscle sends an impulse to make the muscle move. The impulse basically triggers voltage-gated calcium channels to bring in extracellular calcium into the nerve, and that calcium will facilitate or enable that mechanism that allows the acetylcholine to be exocytosed. It basically triggers the snare proteins to become active, and boom, we exocytose from the synaptic vesicles acetylcholine. The acetylcholine goes across that kind of a synapse, which we know is the neuromuscular junction, on the motor end plate, in very close proximity to this area. And again, it's a gel-like area. It doesn't mean it's not like it's an enclosed area with a boundary around it. I mean, it's still tissue that's bathed in what we call interstitial fluid, the generic fluid that's throughout any of the tissues in our body. So what happens, the acetylcholine then goes across that to the various receptors on the motor end plate. It contacts ligand or chemically gated channels, the chemical being acetylcholine, causes them to open. It allows sodium to rush in. 3 sodium ions to every 2 potassium ions that go out theoretically through the same receptor, if you want to look at it from the way the diagrams and the illustrations and animations do things. And at that point, we're going to basically create end plate potentials, localized potentials, which means they don't move very far and they're limited, assuming that there aren't a sufficient amount of them to reach what we call threshold and trigger the action potential. The difference with that is the action potential means that we have effectively opened enough of those channels over a wide enough area at that neuromuscular junction, at that motor end plate, to come in contact with an abundance of voltage gated sodium and voltage gated potassium channels. That means that threshold has been reached, and that means that we will generate an action potential, which is effectively self perpetuating. Once it starts, it will continue and just go and go and go from one to the other to the next to the next, just as any waveform was. Just if you dropped a rock in the water, or a stone that you threw and watched the ripples, they're going to go until they stop. So effectively, that's what you're seeing. So that is effectively the events of the excitation wave, and what happens eventually once that action potential occurs. First, it will trigger this abundance of voltage gated sodium and then voltage gated potassium channels. It goes in all directions. We don't have those inactivation gates that are active that we see in the nervous system to ensure that we only have unidirectional activity. So it'll go out wherever that motor end plate is, it'll go out in all directions. What also helps that tremendously is this relationship between the T or transverse tubules, which are those sort of tangential or right angle extensions of basically the sarcoplasmic membrane, effectively the cell membrane. So that sarcoplasmic membrane or sarcolemma has those sort of interestingly constructed right angle or transverse tubules that surround the individual fibrils. Remember, we're inside of a fiber. It's got those bundles of protein rods called fibrils. When we pull out the fibrils, then inside that we have the filaments like the actin and myosin, troponin, all that jazz that goes on. And so and effectively that wave will be brought to the interior surrounding all the fibrils within the fiber itself, and voila, as it moves, it will form triads. Those T tubules will, on either side of it, along that A and I band junction, you might recall, triggering now again voltage gated calcium channels, but this is an extracellular calcium. This is intracellular calcium. That intracellular calcium will flood the interior of the fiber. It will flood the interior of that particular fibril and induce those changes in that filament filled with sarcomeres. And that effectively is the concept of excitation, starting with the nerve and its voltage gated calcium channels, except that's extracellular calcium, all the way in until we finally release that stored calcium in the intracellular calcium in those terminal cisterns, in those on either side of that T tubule, creating that triad along the A and I band junction, and floods the interior. And voila, what happens then? The calcium bonds with the troponin, that alters its shape. Effectively, that plug now effectively releases the tropomyosin, and it will uncover those active sites on the actin. They're in such close proximity that they're to the binding sites on the myosin heads, which are already preloaded with energy, what we call precocked or ready to go. And boom, they immediately bond. And then that whole series of events occurs where they pivot, and effectively uh, the actins are moved toward the midline, okay, increasing the degree of overlap that's there, and effectively increasing the amount of the A band, reducing the amount of the I band as they're being pulled to the midline. The I band reflects that because the actin filaments are attached firmly to the Z discs, the boundary. The myosin are attached to it as well by way of the titin, but it's less impactful. It doesn't really reduce visibly that that I band, that light band. And the A band just gets effectively becomes continuous and throughout. It doesn't have that sort of midline gap that begins to disappear. That's so-called M or mid zone. You can see all that. Just go over them point by point by point. In the PowerPoint, you'll see the whole thing. So remember all of those factors. We know what an action potential is. Okay. The calcium plays a big role in getting the nerve, obviously, to fire, and it plays a big role once we're inside the muscle fiber to make the contraction occur. And so remember next, the acetylcholine is ligand gated channels. We don't get to the voltage gated channels yet. That first thing of the acetylcholine is a ligand gated channel or a chemical gated channel. We know how that works. Remember the three ways the acetylcholine disappears. It can diffuse away. It can be broken down or metabolized by the very important. You got to know about the acetylcholinesterase, what it does, what nerve agents like acetylcholinesterase inhibitors, those chemical weapons that so-called nerve gases will do by they prolong that as long as acetylcholine there, that muscle will continue to fire. It won't relax. That's a kind of spastic paralysis. So imagine you're inhaling and you can't possibly exhale. It's not a very pleasant way to go whatsoever. So all of those kinds of events are important. This is the kind of material that you're going to see. So what happens at the motor end plate? What does the neurotransmitter do? Yeah, after all of these events, and once the acetylcholine is metabolized, it breaks up into acetic acid, which can be used anywhere and diffuses away. The choline undergoes reuptake. So all of those things that get rid of neurotransmitters, to some extent, there's a little sort of a introduction to that activity by way of acetylcholine when we get to the rest of them in the nervous system proper, which is the next chapter that we really covered in earnest. Okay, so the myosin, remember the attachment with the titin, that's how it attaches. I would call it more indirectly than directly. The titin are those elastic things that attaches its way to the Z disc and the sarcomere. It contributes to the elasticity and the extensibility, so to speak, how you can stretch it and it'll come back, of the individual, not just the fibers, but the fibrils and the filament level at the sarcomere level itself. Okay, the A band is overlapping, myosin and actin. Effectively, it's roughly a 2-to-1 ratio in skeletal muscle and smooth muscle. It's about 12 or 13-to-1. That's why we call it smooth muscle. It's got striations. We can't see it. Okay. A lot of things, we give it a name because the anatomists then didn't have any of the technology. They didn't have electron microscopes, obviously, or high-level scanning and a variety of different techniques that we have today. We know that there are thin and thick filaments that overlap. There are striations. The trouble is, can you see them or not? And so that's where they were named. I don't know that this is how the basis of anatomy, and it's part of what we teach. So the A band is due to the overlap. We know the role of glycogen plays such a pivotal role in that system. Okay, again, with fight or flight, we revisited that. It's stored in the liver. It's stored in skeletal muscle for emergency activity, which we do with some frequency just by doing, even just getting up and running someplace and then coming back, maybe running, you have to go to the store, come back, run. When you do those activities, effectively, it's not fight or flight per se, but you're going into a more active role. So you're going to use some of those stored energy. And as soon as you stop, you're going to replenish it. Remember the idea of staircasing? It's not all or none like a nerve. If we re-stimulate the muscle quickly before the calcium is actively transported back in to those terminal cisternae, they're sort of well-like structures in the smooth endoplasmic reticulum within that fibril, what happens? We have more calcium. We get a greater degree of contraction up to a certain point. So we call it staircasing. The more rapidly we do it, we'll come to something called unfused tetany and then tetany itself. So if you do 10 of those a second, the muscle completely go back to its resting state. If you do 20 a second, you're going to see the staircasing. It's going to increase the degree of contraction. If you do 30 of those a second, you're going to get unfused tetany. If you do 50 of those a second or 70 of those a second, you're going to get fused tetany. That is going to just be, boom, is going to go to maximum contract and stay there. Again, this is more of a laboratory thing, and we use it more therapeutically with regard to electrical stimulation. Remember the action potential, and this is true for the nervous system. Effectively, what creates depolarization? Sodium. Remember the first thing we encounter are voltage gated sodium channels, sodium rushing in, increasing the resting potential from its whatever negative 55 up to maybe positive 30. Okay. And on the flip side of that, the exiting of the potassium, bringing that back down, not as rapidly, but fairly rapidly, is what repolarization is about. So it's important to remember both of those. So repolarization is mostly due to allowing the potassium to exit the cell. Depolarization is allowing the sodium to enter the cell by way of those voltage gated channels. I think that's really important. So you should understand all of those. Don't forget the 3-to-2 ratio of sodium coming in, potassium going out, and understand we reverse that ratio, so to speak, by the sodium potassium pumps. So that should be part of what you recall about this chapter as well. Let's see, I covered that. I'm just telling myself. Remember, A bands are dark, I bands are light. Please don't forget that. Drive me crazy. Okay. We talked about what happens with additional calcium, energy usage. Okay. Effectively, remember, we have a limited amount of ATP. When that's used up, the next thing we use is that creatine phosphate. And effectively, it's another source of high energy phosphates. And obviously, that's important. And after that, I was just thinking what's coming up next. After that, we go into anaerobic metabolism. Obviously, there's a limit, but what is produced is lactic acid. There's other things that are produced in the sense that we get irregular concentrations of charged particles, uh, electrolyte imbalances, and effectively that leads to cramping in the muscle and loss of function in the muscle for sure. But the big waste product that sort of underlies that, even though we can utilize it elsewhere, is lactic acid. And obviously, when we stop the exercise because we physically can no longer do so, eventually that lactic acid is converted by the liver to pyruvic acid so we can effectively recycle it back by way of the Krebs cycle. The secret sauce for that is the oxygen, because what caused you to stop is that stupid acronym, that EPOC. We always call it catching your breath. That exercise post oxygen consumption is what that acronym is. So that's anaerobic metabolism. When you're doing aerobic metabolism, it's aerobic. Okay, you're basically, think of what you do when you're working out at the gym. You increase your activity aerobically. You're on a treadmill. Maybe you're jogging. Maybe you're rapidly walking. Maybe you're on the cross trainer, whatever. And you increase your respiration to blow off carbon dioxide. And at the same time, you release water by perspiration as well as cooling yourself. That's kind of how it works. Okay. So all of that type of stuff is important to remember. Remember the idea of that golden zone of 80 to 120 for optimal sort of distance. So if the muscle is too far stretched, effectively you're not going to have enough potential areas for those cross bridges to form to generate any power. If it's already mostly contracted, again, you're not going to be able to, where does force come from? Mass times acceleration. You can contract the muscle some, but you have very little distance, and you obviously can't do so very rapidly if it's already contracted. To sort of apply your physics and what force is, mass times acceleration. Okay. And so either we can't generate the acceleration because it's already shortened, or we don't have enough contact to mechanically, generate enough contact areas to sort of start the process going. That's kind of what it's like. So whether it's stretched beyond 120 or well below the 80, if it's already contracted, that's the problem area. So that's really part of that has to do with muscle tone, and we try to maintain it in that direction. Okay. So let's see what else. All the areas. Remember, actin's a very interesting one. We've talked about actin a lot. In the cell, the cleavage furrow, so it's active in mitosis. It can construct and reconstruct. It's all about forming pseudopods and amoeboid motion and assisting with endo and exocytosis. That's actin. Obviously, it's pivotal for muscle contraction. Okay. It's pivotal for mitosis. It's pivotal for amoeboid motion. It is so important, and that's the beauty of it, because in its globular form, it's soluble, so we can deconstruct and reconstruct. But when it's put together in its fibrous form, like we see it in fibrous actin, in vis-a-vis actin and myosin filaments inside a sarcomere, it's insoluble and therefore can generate strength. It's a very, very interesting molecule. But it's very important to note when you see that's another student confused things. If it says G actin, it's globular, soluble. If it says F actin, it's not. Okay, that's now we're into that F, that filamentous type structure, which by definition is insoluble. Okay. Uh, Remember all those voltage gated channels and things like that? If they're in the nerve, they're basically along the membrane, the external membrane of the nerve. If we're talking about in the skeletal muscle, it's along that sarcolemma. Effectively, it's along the cell membrane. So is the T tubule. All of those are where you find voltage gated sodium and potassium channels. That's really an important thing to remember as well. Okay. Uh, a few things, and then we get into the nervous system per se. You have glial cells, all there are 6 of them, 4 for the central, 2 for the peripheral. Heavens, they're easy. The 2 for the peripheral are similar but different than the ones in the central. 2 for the peripheral are the satellite cells that we think, and I emphasize the part think, act like astrocytes, which are the ones that are the most versatile and certainly the most abundant in the central nervous system, and the Schwann cells, which act like the oligodendrocytes making the myelin sheath. The difference is the construction, where because you have limited space in in the skull and in the brain, that you have the myelin sheaths. You're going to have one cell is probably going to gather, as many as 60 different axons, where in the peripheral nervous system, you have an abundance of room. So the Schwann cells are sort of like individually wrap along those very, very long, sometimes even a meter long axons and intervals. Okay. So you just look at the diagrams, you'll see the difference. Even though effectively their membrane is myelin, that sort of fatty and proteinaceous insulating, the thicker the myelin, the thicker the nerve, the more myelin, the faster it works because effectively it insulates. And those gaps, whether you call them nodes of Ranvier or myelin sheath gaps, are where the abundance, that's where the sodium, voltage gated sodium and voltage gated potassium channels are located. And in this case, the voltage gated sodium ones have that little inactivation gate. That's a big player because this is when we start to see absolute refractory period. It's not as pivotal in the heart where it plays such an important role in limiting how rapidly your heart can beat. Here, the big thing is that plays a really significant role in the unidirectionality. It's why we don't have it going where that signal is going to go from where it's stimulated toward the termination, toward the axon. Okay, from where it starts to where it ends, it can't go backwards and forwards, that type of thing. So, or in multiple directions, like in muscle cells. So we have that sort of unidirectionality. The inactivation gates deactivate, okay, cause that absolute refractory period. So only downstream can that impulse proceed, not backstream, not upstream and downstream simultaneously. So please remember that. The astrocytes play an important role in the blood-brain barrier. It's mostly due to those tight junctions, but all those cells are covered by those astrocytes. Nutrients and waste products, oxygen and carbon dioxide, almost pass within and along those membranes of the astrocyte as well as the capillary and into the nerve itself. Okay, so there's a really pivotal role, and that's what it's all about but small molecules that are fat soluble being so important both pharmacologically and and things that we make within ourselves that have the capability of crossing the blood-brain barrier. Okay, I think that's part of the microglia, or little scavenger cells. The ependymal cells play an important role in circulating and to some extent finalizing that sort of plasma base, but not the same as plasma material we call cerebrospinal fluid. So, know all the names of those glial cells, whether they're central or peripheral. Okay, know what the blood-brain barrier is, and I've just described it. Okay, where does the myelin come from? The Schwann cells, PNS, the oligodendroids, CNS. You're getting the idea. And then we get to smooth muscles, like I talked about before. There's a greater amount of thin to thick. So even though there's overlap, we don't see it, so we've taken to calling it smooth. It's very different. They don't have the same equipment. Okay? Even though it has intracellular calcium, the calcium comes from the outside. It doesn't have T tubules. Okay? It doesn't have troponin. It's got tropomyosin, works similarly, but not quite the same. Okay? It doesn't have a neuromuscular junction. It has those little varicosities. Nerves go through it. A varicosity is sort of what we would call a bulbous enlargement. Okay? And if you look at you can see the illustrations, and the neurotransmitters come from that. And it's not only acetylcholine, though that may be as frequent as any. Okay? So it's not limited to that. Okay? So I think you need to be pretty familiar with all of those things. Right? Let's see. And the difference of calcium, really, I have to continue to underscore that. It relies on extracellular calcium, even though there is some intracellular calcium that certainly plays a role. And it doesn't bond, like it's different. We don't have those quote unquote specific structures like troponin. It bonds to something called calmodulin. The calmodulin, once it's activated, activates those myosin kinases, and that's really what triggers the eventual bonding of the actin and the myosin that we see there. So we activate that myosin kinase, and that facilitates the cross bridge, better way to express it, between the actin and myosin. Okay, they don't have Z discs. We call them dense bodies. They're sort of bulbous in the middle, and the structure is somewhat spiral. So it's almost like diagonally or lattice-like. So when they contract, they don't contract. We don't get that sort of shortening that occurs with the sliding filament theory. If anything, you almost get this sort of spiral kind of bulging in the center contracture. They're organized because remember, you have principally first circular around the lumen and then longitudinal. That's a consistent pattern. We'll revisit that in a variety of systems. Smooth muscle is not as powerful. It's more about endurance. It's interesting to me. Power is from skeletal muscles. Endurance is from with limited with strength, but not dramatic contractures, like we're thinking about lifting a weight or moving a heavy object that we see in skeletal muscle. And to me, the heart's sort of the interesting one in the middle, though we do heart separately, because that's got a tremendous amount of power and a tremendous amount of endurance. So it's a little bit of both. It's got kind of the best of both worlds that are there. Okay. I think that's all important. I commented on the downstream direction and really those inactivation gates playing such a pivotal role that are there. We know about the resting membrane potential. I think that's really important. And again, there's a big difference when we talk about we don't have end plates, so we don't have end plate potentials. So end plate potentials are not sufficient to get to threshold. It's a little bit different language. We have different kinds. We have those, sort of graded, they call them potentials. And because it's not in the neuromuscular system, everything is positive. We're going toward threshold. In the nervous system, a nerve can communicate and say fire or don't fire. And frequently the same nerve can say both of those things, not simultaneously, based on a variety of frequencies that it receives on its own impulses. So effectively, we call those graded potentials either depolarizing or hyperpolarizing, taking you closer to threshold, depolarizing, more positive, less negative, more positive, hyperpolarizing, increasing the degree of polarity, more negative, less positive, actually increasing in a negative direction. That's important. So know which neurotransmitters and how that kind of works. Typically, if we open chloride gated channels, that's typical the way we hyperpolarize. Chloride negative comes in, lowers that resting membrane potential. And typically, when sodium comes in, positive sodium, that brings it, that's more of the depolarizing type agent, bringing it closer to threshold. And there's variation in threshold depending on the nervous circuit. That's there. And so its ability to fire very rapidly is due to that same kind of thing. Okay. Those voltage gated, the presence of the inactivation gates really play a pivotal role in that as well, because you have that absolute refractory period, but there's so many of those inactivation gates there, it really is very significant with regard to it. So both of those, the ability to fire rapidly and the fact that all or none, have to do with the abundance of voltage gated sodium channels and the presence of the inactivation gate. That's a good way to look at it. And let's see. And again, because it's all or none, amplitude is frequency. What makes it, the more it fires, the more rapidly it fires, the stronger the signal we perceive. Hotter, colder, painful, loud, whatever. We talked about that a lot. Okay. Uh, I'm almost there. So know the difference between end plate potentials and graded potentials. I think that's really important. Okay. Let's see. Okay. Oh, the things it's coming to mind. I'm just trying to think of things I might have forgotten. What really accounts for that resting membrane potential consistently? Sodium obviously predominates on the outside part of the membrane, potassium on the inside. Even though their relationship, because of the differences between them, and obviously the abundance of those leak channels, particularly the potassium ones, create that resting membrane potential. What offsets it? Even though we have neutral, okay? And so the extracellular fluid and the intracellular fluid are neutral. Why are they neutral? Very simply, because you have chloride on the outside neutralizing it, and the proteins that are trapped with a lot of anionic, the negatively charged proteins on the interior. Those are the ones that create the zero, so to speak. Okay. They're the ones that create, when I say zero, that it's, even though it's more positive on the outside than the inside, the extracellular fluid and the interior fluid, the cytoplasm, is electrically neutral. That's the best way to say it. Okay. And lastly, there's one question, it's called a sequence question. What's meant by a sequence question? Okay. It's a batting order. Okay. In this case, the batting order, I'm telling you what it is, has to do with the events of a nerve firing. So what happens? We end up with voltage gated calcium channels. They're the ones that open up. They allow the neurotransmitter, just like the neuromuscular junction, for neurotransmitter to go across. Assuming it gets there, I'm assuming it's in a sufficient amount to create an action potential. The action potential, okay, will by definition, the action potential will be of a sufficient distance to contact those voltage gated sodium channels that will open and allow the rapid influx of sodium with the action potential spike of depolarization. Those channels will inactivate by those inactivation gates. Immediately thereafter, the voltage gated potassium channels will open. That will create the outflow of potassium that allows repolarization to occur. And at that point, the sodium and potassium pumps will activate, because the electrical conditions were restored by first the sodium and then the potassium, and now we have to restore the chemical conditions by way of the sodium potassium pump. So remember the order. We have to somehow get to threshold. Okay. One nerve talks to the other, comes down. For the first nerve to fire, we've got to get a voltage gated calcium channel open. Okay. It releases the calcium extracellular calcium, goes in, facilitates exocytosis of the neurotransmitter. The neurotransmitters come across, they generate whatever their potentials, assuming there's sufficient amount of the appropriate type. We generate an action potential. That action potential reaches first voltage gated sodium channels. They create depolarization, the upward spike. The inactivation gates create that absolute refractory period. Then the voltage gated potassium channels open. They create the repolarization back, even with the overshoot, back to its resting potential. And at that point, we have to utilize, in order to restore the chemical conditions inside, if you will, for continuous usage, even there may be limited usage, relying on the sodium and potassium pumps. That's it. That's your review. Nice to talk to you. Have fun. Bye bye.